In this following lecture, we are going to learn about heredity and variation, Mendelian inheritance, Mendel's experiment and its procedure, characters, and monohybrid cross. All living organisms inherit characters from parents to offspring, such as you have inherited some characters from your parents to you. They have inherited some characters from them to you. Ever wondered why puppies look like dogs and kittens look like cats and not the either way? It is because they inherit some characters and carry some part of a gene which encodes puppies to be puppies and kittens to be like cats. Now let us know what is gene. Is this the one? No, that's a gene, not a gene. This gene is what we wear, not what is inside our DNA. So what is gene? Gene is actually a small part of DNA which encodes or stores the information about you, your being. Just like a bank stores money, DNA stores information. Now let us see what is inheritance. Ever wondered why Zayn Malik look like Zayn Malik? Cute puppy is looking like cute puppy, and this butterfly is looking like this butterfly, and why are the bacteriophages so small and they are not like each other or duplicates like they show in movies? It is because of inheritance, some sort of family code which only your family will give to you and not to someone else outside the family. Let us see what is heredity. Just observe these both pictures which is getting showed on the screen. Heredity is something which is passed from one generation to another. Like characters are passed from parents to you. You can see in the picture the son at a particular age resembles the father at the same age. See how heredity is passed on. Same to same. See the grandmother and granddaughter picture you can see here of the same age and in the same pose. See the resemblance, but they don't look same. They can look alike but not same. So this similarity, some similarity which is passed from your parents to you is known as heredity. Variation or the differences or the characteristics which makes you different from your parents are known as variations. Variations which are characters differentiable or different from parents to offspring, unlike this show in movies like father like son. In reality, a little bit similarity can be possible, but we humans reproduce offspring, not clones. Now let us know about Mendel. Mendel was known as father of genetics. He studied pea plants and also he discovered many aspects of genetics and was the first scientist to do that. Because of him, we know all the information we know today in genetics. Laws of Mendel, which he has put forth, is known as Mendel's law of inheritance, which we shall study in this chapter. He studied various aspects of pea plants, such as the height, seed color, seed shape, pod color, pod shape, flower position, flower color, etc. etc. You can see it in here. The seed shape can be round or can be wrinkled. Also, the seed color can be yellow or green. Flower color can be purple or can be white. Pod shape, the shape of the covering in which the peas are coming, can be inflated or pressurized or can be constricted. With flower position can be axial near the axis. See, this is the axis. Or can be terminal. Terminal means at the apex. See. Now next is the stem height can be tall or it can be dwarf. So these are some of the characters Mendel showed. Now why did he only pick up pea plant? First of all, it can grow throughout the year. That means in a year, since there are 12 months in a year, one cycle of pea plant takes 3 to 4 months. That means 3 to 4 cycles are possible throughout the year. Next one is, it is a very small plant, takes nearly very short space to grow. But in that short space, it can grow lots of yields and we can get lots of crops, lots of varieties in merely 2 by 2 square feet of area. Why he selected pea plant is because it was self-pollinating. Now self-pollination is a process in which, see these pollen grains you can see at this point, see these pollen grains. This pistil of the same flower or the female organ collects the pollen grain. The pollen grains go from pistil beneath and goes to ovary. The stamens being the male organ, pistil being female organ. Stamens pass on pollen grains to pistil, and these pollen grains get passed on to ovary where there is development of seed. So that is how self-pollination takes place. Now he studied various characters such as character, trait, factor, gene, alleles, recessive, dominant. We shall know in detail what are they. There are different characters, and Mendel studied and take into accountants, that is characters, trait, factors, gene, alleles, recessive, dominant, etc. We will learn about it further. See, these are the various characters he studied in a pea plant, and that is the reason why he selected pea plant only. First, as you can see here, there are various meanings of the given character. But first of all, if we say about character, we say a character or a feature of an organism. Next is trait, which is inherited character which is received by the offspring through their parents. As you can see here, there is an example of a trait or an inherited character. You can see the similarities between the father and the daughter. They are looking so similar, so that is something which is a trait which is inherited by her father to her. Next one is factor, which is known as unit of heredity. This is the basic unit of heredity due to which things are passed on from parents to, this is responsible, our DNA or the factor. Now next one is gene, which is known as segment having genetic information, which encodes the genetic information. You have already seen what is gene. Alleles which are known as substitute of gene. They are varied types. Allele is something which we can say that it is subforms of gene, that is the height can be long as well as it can be short. It is known as recessive gene or the gene which is suppressed. An example can be given as eye color. Normally we Indians have brown eye color, but can find people having lens of different colors. The brown color is said to be dominant trait, while these grey, blue, etc. types colors of the lens are known as recessive. These are due to recessive gene, and the brown color of the eye is due to dominant gene. So we will move on to directly phenotype or outer appearance. We have already covered recessive or dominant. Then phenotype, or it is basically how an individual looks from outside, and the basic appearance which we see from the eyes, that is the basic structure, shape, by directly watching them. Next one is genotype or genetic makeup. Genotype means the constituents of the gene, like the basic recipe of the genes which are present inside an individual, and phenotype, it is what we see from outside. See, we are going to learn about homozygous and heterozygous together, because homozygous is something which is known as when an individual possesses similar alleles for a particular trait is known as homozygous. As you can observe here, the homozygous, there are capital A and capital A alleles present in this individual, hence it is known as homozygous. Now what is heterozygous? Heterozygous is a condition in which individual possess dissimilar or different genes, different alleles for a particular trait, like small a and capital A. One would be recessive and other one would be dominant. So that is known as heterozygous. Now what is known as pure line breed? The pure breed with whom the cross is began like taking a homozygous trait. Now hybrid cross. Hybrid cross is something which we can get by crossing of two parents, and we can get a new hybrid. For example, this is the example of hybrid when a tiger and a lion are crossed and we get liger. So that is something which the parents are different and the genetic makeup is different. So when they reproduce, their offspring is something which is known as hybrid. Next one is Punnett square method. We will learn about monohybrid F1 generation, F2 generation in further lecture. So don't worry about that. Punnett square method is basically the graphic representation of the cross we do. It is very helpful in studying how much is the progeny produced, what is the genotype and phenotype ratio, and you will learn about it in future. Now let us know what was the procedure of the experiment. Mendel was very methodical. He first considered only one trait, did a monohybrid cross. Then slowly and steadily he took more traits and made dihybrid cross. He considered trihybrid cross, but he made sure he started with pure lines only, that there were no hybrids in the parents from which he is starting the experiment from. He also maintained many records of his experiments. Suppose the parent A and the parent B. Parent A bears, which is homozygous allele for tall, and capital B is a homozygous dog parent. So he took these parents only, so that there are no hybrids involved, and we get the hybrids in further generation, so that there is no mixture in between. He started with pure lines. He artificially selected these parents. He crossed the selected parents so that there are F1 generation, and he get the desired trait by doing emasculation. Emasculation is basically a technique which is known as removal of stamens, but the stamen is basically male parts. The anther, filament and stamen as female parts are concerned. The stigma and the style make up female parts. So what happens here is, there is a A parent and there is a B parent. He selected the A plant to be female and the B to be male. Now what happens is, since the flowers have both the parts, that is pistil and stamen, so when he has selected A parent to be female, he would remove out stamen by plucking. So there would be only pistil remaining for the A parent, and so automatically the plant would be female. Now in B parent, he plucked out pistil part, so what would remain? Stamen part, the male part, and this stamen is rubbed off on the pollen from the selected male is rubbed on the stigma and style, or the female organs is allowed to produce a F1 progeny. In short, we selected a plant to be female and the B plant to be male. The A plant has a characteristic for tallness. We selected this as this, capital T, and the B plant, or the male plant, to be dwarf. There was cross between them. Female parent gave capital T and capital T. Male parent gave small t and small t. After hybridization, what happened was, this one gets linked with this and gives capital T, small t. So as you can see here, the four offspring produced are monohybrid tall. He self 2 hybrids, and what he received was Hey, thank you for watching the video. If you like the video, don't forget to comment, like and subscribe. And if any doubt, you can email us at the given email address below.